The overall goal of this procedure is to generate dispersed cultures for long-term imaging of oscillations in fluorescence reporters of cyclic gene expression in single isolated cells from the zebrafish segmentation clock. This is accomplished by first dissecting presomitic mesoderm or PSM tissue from transgenic zebrafish embryos. Next, the most posterior piece of the PSM, the tail bud, is dissected from multiple embryos and the pieces are pooled in trypsin. Then, the tail bud pieces are manually dispersed with a pipette, generating a cell suspension. Finally, the cell suspension is plated onto fibronectin-coated culture dishes for long-term imaging. Ultimately, results can be obtained that show multiple oscillations in intensity over time from single PSM cells through fluorescence time-lapse microscopy.
The main advantage of this technique over existing methods such as in situ hybridization of gene expression in PSM tissue is that our fluorescence reporter allows for real-time monitoring of the dynamics of oscillations in the segmentation clock at the level of single cells.
This method can provide unique information to the segmentation clock, but it can also be used in other systems like organogenesis or disease models, where it can give us information about behavior at the single cell level, where the appropriate transgenic lines are available.
On the day before dissection, obtain embryos from an in-cross of zebrafish pairs heterozygous for the transgenic allele. Raise the embryos in E3 medium without methylene blue at 28 degrees Celsius until shield stage, about 6 hours post-fertilization, or HPF.